Welcome everybody to this evening's lecture. My name is Janice Reimer and I'm the Dean of Undergraduate Medicine, and this is the inaugural lecture of what we're calling the Research Stars, because as a medical student you come to medical school and you know about education, and in the clinical years you know about service, but often what you don't realise is that you're in the presence of greatness. So what we wanted to do was to pick people in the medical school who we thought were outstanding at research and ask them to come and talk to the medical student body about their research. So it gives me great pleasure to introduce Lucilla Poston, who is head of the Division of Women's Health and also is the director of the Maternal and Fetal Research Unit, which has a multidisciplinary team which focuses on translational research. And Professor Poston's own research interests are the developmental origins of health and disease and preeclampsia. And it gives me particular pleasure to introduce Lucilla because I'm an obstetrician and gynecologist, and Lucilla was my previous head of division. So Lucilla, I'm really grateful for you to come this evening to talk to the students about your research and entitling your lecture, Pregnancy, A Window for Health for the Next Generation. Thank you very much.
Thank you. Well, good afternoon, everybody, and thank you very much for turning out in such wonderful numbers. It's brilliant to have so many people here. I must say, when I got the email from Janice, I thought, stars? That's not me. And then I thought, well, actually, maybe it's not so entirely inappropriate, because I see stars daily, but they're not my own stars. I think what we have to say, you know, those of us who are heads of division and so on, is that the stars of research are those people who do the work at in the lab, the people who go and recruit the patients in the clinics, and so on. As head of division, alas, we get very little time to do our own research, although it has to be said, I really try hard to keep on doing it. So what I'm going to talk to you about today is not just my own work, quite a few of my ideas, and working with a wonderful group of collaborative people. And what I'm going to talk to you about today is about pregnancy. But it's about the suggestion now that pregnancy is incredibly important for the health of the next generation. I think we've always known that, but there are new hypotheses which suggest now that it's just more important than we ever originally thought. So I'm going to give a little vignette about a new hypothesis, one which may have crossed your path or it may not, but I think in all the years that I've been in research, this is probably the most exciting time. And it's because it's a hypothesis which has taken a while to gain mainstream recognition. It's one which is potentially incredibly important for public health, and one where there is a very, very good evidence base now, whereas 15 years ago I think we wouldn't have been able to say that. So if we just think for a few minutes, I mean, even the first years amongst you will know a bit about risks in pregnancy, because everybody is affected in some way. They have a relative or someone who's had a problem in pregnancy. So the fetus usually is fine, and pregnancy outcome is perfectly okay, but in a significant minority of women it doesn't quite work out, does it? And I think we've all known people who've had premature deliveries, premature labour, and the babies are threatened because they're born with immature lungs and they have a high risk of infection. And indeed, if they're born very, very prematurely, a high risk of death. And we all know how sad that is for families. One area I'm particularly interested in terms of maternal problems is the disease preeclampsia. And that is, comes out of the blue. Preeclampsia is a prodrome to eclampsia, which women can be affected by epileptic-type fits. They can get thromboembolisms. They can die. It's extremely serious. The babies can die too. And that's a condition that affects about 4 in 100 pregnancies. That's another common one. There are some more esoteric problems, such as septic cholestasis, where women's livers become affected, and so on. And for the baby, there are risks too. If the placenta doesn't work properly, the baby is subjected to a reduction in nutrient availability, and the growth-restricted baby, the growth-restricted fetus, has a high risk of early death and sepsis and so on. And then we know about the effect of teratogens. I mean, thalidomide always springs to mind as being a drug which was given for all intents and purposes to improve pregnancy outcome and help mothers who are being sick, and we all know the problems about that. So I think we're all, in our everyday life, come across problems that women face in pregnancy. But that's not essentially what I'm going to talk about this afternoon. What I want to share with you is the suggestion that there is a more insidious, more silent effect of the environment in utero, which only becomes apparent in later life. So not the immediate effects of prematurity or indeed the morbidity associated with that, but more subtle influences of the environment, particularly the nutritional environment, which leads to an enhanced risk of disease in later life. Now, the guy who really brought this into the public arena was a chap called David Barker, who was a professor at Southampton University, and it became known as the Barker hypothesis. And what David Barker suggested was particularly on the undernutrition side of the nutritional spectrum. So babies who were born small because they hadn't got adequate nutrition or blood flow in utero. Now, what happens to those children? We know a lot about the physiology of in utero growth restriction. We know, for example, when a baby has a choice to make about which organs it's going to perfuse, if it has little nutrient availability and oxygen and so on, that the baby's blood flow is directed to the organs which really matter, to the brain and to the heart and so on. You get distribution away from the periphery, away from skeletal muscle, and away from the kidneys. And it stands to reason, therefore, that it isn't rocket science to think that that might have some persistent influences on the metabolism and the risk of disease in later life of that child. And those are adaptive mechanisms. Those are mechanisms which help the fetus survive in utero. They're good. But in fact, the suggestion is that the baby is born into a plane of nutrition which is unexpected. In utero, it's expecting it's developed an adaptation to undernutrition, and then it's suddenly subjected in postnatal life to a relatively hypernutritional status. And that hypothesis was also known as the thrifty phenotype hypothesis, and developed with David Barker by a guy called Nick Hale, sadly now dead, who was in Cambridge. And then it changed a bit over the years, and it became known as the fetal programming hypothesis, and is now known as the developmental origins of health and disease hypothesis. And essentially what David Barker said, and I'm going to show you some of the original data, he suggested those babies who were born small and hadn't attained their growth potential developed cardiovascular disease in later life, developed dyslipidemia, developed cardiac dysfunction, and were more likely to die early. That obviously is not the only cause of those disorders, but quite a potentially important cause. And he had lots of good epidemiological studies to back up that supposition. So that was in the 1990s, and actually is when I started becoming interested in this field. And in fact, the reason I started doing this work was because I didn't believe it. I went to one of David Barker's talks, and it was at the International Society of Hypertension in the early 1990s, and he was practically booed off the platform. And unfortunate, because a new hypothesis is often met with derision. I think we all have to appreciate that. You have to be bold, you have to stand up for what you believe. But it seemed to me and to a lot of other people to be intrinsically unbelievable. But what is it now, 20 years later? It's now a mainstream hypothesis. The World Health Organization, for example, has taken on the developmental origins hypothesis and is recommending that one thinks about life as a lifelong course of health, starting right at the beginning of pregnancy, not when a baby is born. The Royal College of Obstetricians and Gynecologists has got a paper on life course of health, which includes the suggestion that we should look to pregnancy as the beginning of the start of health. And there's an eminent society now, which I sit on the council of, which has meetings with thousands of people attending, the next one being in Singapore next year, and people have written books and so on. It's become mainstream thought. So what is the sort of mechanism? Those scientists amongst you, and I'm a scientist by origin, will be wondering what on earth is going on mechanistically. And indeed, it's the mechanistic side of this which I've enjoyed doing, and I'm going to show you some data that we've got from animal models, which has really helped convince me about this whole scenario. And essentially what is proposed is that the fetal genome is susceptible to environmental influences in utero, and through epigenetic processes, and I'm sure many of you have heard about the influence of environment, the nurture-nature interaction. There is an epigenetic process which either acutely or in the long term leads to the exacerbation of chronic disease in later life. And I'll just take you through a little bit of what we think might be going on. Now, those of you who don't know much about genes are no, I don't know very much about genes either. I certainly work with them. But here is a gene, and here is a transcription factor binding on, and when the transcription factor binds to the gene transcription occurs and everything is set off. We all know that. The transcription factor binding can be affected by the methylation status of the promoter region of the gene, as shown here. So changes in binding of methyl groups can reduce the binding of the transcription factors, and this gene is essentially switched off. And that demethylation process is incredibly important in normal embryonic development. Imprinted genes, as they're called, the methylation status changes completely during development. Alternatively, the histone tail of a gene is important in terms of gene expression because the histone acetylation is very important for binding of transcription factors. So what is proposed now in this field, and this is where there's a lot of good biochemistry going on now, is that acutely perhaps the epigenetic processes lead to a change in development of a particular organ, for example in the brain or in the liver. Structural changes then ensue, and then they persist into later life and cause the problems. Alternatively, some suggest that the methylation status of a gene can be persistently altered, and that's slightly more controversial, but there's some evidence for it. And that leads to that gene being either switched on or switched off in later life, and that can be tissue-specific, which is very important. So this has got into the public domain. This was a paper that was published last year in Diabetes by a close collaborator of mine, Professor Keith Godfrey from Southampton. And it got picked up by the Daily Telegraph, and what they said was that obesity DNA is triggered by a poor pregnancy diet. Well, what does that mean? And they say women who eat badly during pregnancy can change their children's DNA to make them more susceptible to obesity, scientists have discovered. Well, actually, what the paper said was slightly a little more esoteric than that, which is the epigenetic gene promoter methylation at birth is associated with child's later adiposity. And in fact, what Keith and his colleagues had found was that the methylation status of the retinoid X receptor alpha gene was associated. This was taken cells from the umbilical cord. He looked at the methylation status of the gene in the umbilical cord, and the methylation status was proportional to the child's fat mass, expressed in different ways. And what they also found was that this was associated with the carbohydrate content of the maternal diet, hence the Telegraph picking up this was mum's diet causing changes in obesity predisposition. It's actually a long way from proving it, and this was a sort of proof of principle study but attracted quite a lot of attention, and there are many of them out there now which are proving increasingly that there is something to this persistent change in methylation status. A more acute sort of demonstration, this is very much a proof of principle study, but it demonstrates very nicely what I'm saying. There is a sort of yellow mouse called the agouti mouse, and it has the agouti gene, the AVY gene. And that gene is very susceptible to being methylated or demethylated. The methylation is on-off. And when a yellow mouse, a yellow agouti mouse, is mated with an ordinary brown mouse, you get the offspring with different colours of their coats. So that's what normally happens. But if you supplement the mother with folate, which changes the methylation profile, it's folate involved in the 3-carbon cycle, this study showed that you get a different distribution of these different coloured coats. So the black bars are those animals which have been subjected to maternal folate supplementation. You basically get more of the darker coloured coats. And they looked at the methylation status of the agouti gene in these animals, and this change in the phenotype of the offspring was associated with changes in methylation status of the agouti gene. It's quite a nice demonstration. It's not really developmental program as we understand it, but of course folate is given to pregnant women to prevent nucleotranslucity defects and so on, and we've got to think about what happens if women overdose on folate. I think there is a potential problem there. In fact, we're about to start a study next year where we're going to be giving quite high folate supplements to pregnant women to try and prevent preeclampsia, and we shall be looking very carefully at the methylation status of some relevant genes in the children. But to go back to David Barker's original work, and I just want to put this in perspective and show you some of the original data which was so groundbreaking at the time. he was very good at picking up mother-child cohorts around the country. He did some work in the north of the country, in Preston, and he picked up a cohort in Hertfordshire where the midwives and the doctors had very carefully collected lots of data about birth weight and placental weight and all sorts of things, and they were also able to follow up those children when they became adults. They all seemed to stay around in Hertfordshire. And so one of the first studies was this, which was published by Caroline Fall, who is one of the major players in this field. She's actually now working on this hypothesis in India, and showing an association between being born of low birth weight and systolic blood pressure. Now, the importance of this is, I'm sure you're all saying, I bet there are genes which predispose the mother and the child to high blood pressure or to diabetes and so on. And indeed, there are, and I could give you chapter and verse. Several studies have looked at the genetic predisposition to these various phenotypes in the offspring, and it seems to be unrelated. So when we say there's an independent relationship between low birth weight and systolic blood pressure, what we actually mean is that the statisticians who've done this work have adjusted these associations for known confounding variables. Now, what we mean by that is socioeconomic status, genetic susceptibility if it's there, they have the data, educational attainment, the BMI at the time of study, and so on and so forth. And by teasing out the relative components of the in utero environment, they can say with some certainty, but certainly not with complete certainty, that there's an independent association between being born small and these offspring characteristics. And it is not proof, and it's extremely difficult to prove it. Another very well-quoted example is the story of the Dutch hunger winter, which was an unfortunate natural experiment. Towards the end of the Second World War, actually the last winter of the Second World War, the Nazis besieged the southern cities of Holland, and particularly Amsterdam, and they basically prevented food getting to the population for a few months. And in the same time, all the obstetricians and midwives carried on collecting records from the mums about the birth weight and the exposure to the famine and so on, wonderfully detailed records. And the group in Amsterdam is still working on the children of the Dutch hunger winter, as they're known. And they've produced the most incredible series of papers. There's someone called Professor Rosenboom who's done all this work. And essentially what they have found is that if a woman is subjected to supreme undernutrition in the early stages of pregnancy, they've looked at different trimesters, and they've looked at the relationship between the BMI of the offspring, the blood pressure, cardiovascular risk, and so on. And there are very strong associations between maternal undernutrition of this extreme nature and the risk of disease in later life. In fact, I know somebody who's in his 70s now who was a victim of the Dutch hunger winter, and he has one or two nasty complaints. And I mean, one never knows, but he always attributes it to the time when he was in utero in the Dutch hunger winter. So there are hundreds and hundreds of papers written about this. It's a complex literature to get through. But since those days, many studies have shown a very nice systematic review was published only a couple of weeks ago, again showing this association between being born of low birth weight and failing to achieve growth potential and hypertension in later life. And it seems that the problem largely is with systolic blood pressure rather than diastolic blood pressure. And it's a modest effect, but it seems to come through loud and clear in nearly every study. And others have looked more intensively at cardiovascular function, for example, using ultrasound to look at brachial artery endothelium-dependent dilatation, and shown low birth weight children, when they grow up, have increased problems with endothelial dilatation, with arterial stiffness, so on and so forth. And if you read this literature, it is really quite convincing. But the problem is, all these cohort studies are association studies, and by their very nature, epidemiological studies of that kind can infer causality, but they can't prove it. And so a number of us across the world, in fact many, many different laboratories, have turned to animal models to address the developmental origins hypothesis. And of course, one can control the environment of an animal. They're all genetically identical if you use inbred strains. You can use knockout animals, which some people have used and which we're using at the moment. People have shown across the species that this phenomenon occurs. So people working on baboons, for example, have shown that undernutrition is associated with high blood pressure in the offspring. People have worked in the Japanese macaque in Portland and Oregon and shown the same. Many studies using sheep, and the fetal sheep is a very good model to investigate fetal physiology. That is actually how, using fetal sheep, that it was discovered that glucocorticoids can cause maturation of the fetal lung, and that is why all women with threatened premature labour now are given glucocorticoids. That was discovered in fetal sheep studies, and it's a very well characterised model. And then others, including ourselves, have used rodents, rats and mice. So just to stick for a moment to the undernutrition side of the paradigm, one can reduce blood flow to the rodent fetus by ligating the uterine artery partially, or in sheep you can remove a bit of the placenta, a caruncle of the placenta, to reduce blood flow. Others have subjected the mothers to undernutrition to rather sort of mimic the Dutch hunger winter, or indeed the diet in developing countries. And universally, all of these studies have shown that in very well controlled conditions, when the fetus has growth restriction as a result of these interventions, that the offspring develop hypertension. And this is a study that we did many years ago when I was working with Mark Hansen at University College. And Ozaki was one of our PhD students. He was a Japanese research fellow. And he did a very nice study where he reduced the diet of a rat just by 30%, so it's 70% of the normal diet. And we followed the blood pressure of the offspring up. We followed a lot of other things, but I don't have time to show you all the data. And as the animals grew up, it was interesting. We often see this, that the males were particularly badly affected. And we measured mean arterial pressure by using an arterial probe. This was very accurately done. And as the animals got older, just through exposure in utero to a very modest reduction of maternal dietary cost of the broad diet, these animals grew up with increased blood pressure. And this is what so many people have seen across the world. So one doesn't really believe those human studies because of all their potential drawbacks. This has been shown time and time again. And we now have some very good insight into what the mechanisms are. This is not just observational physiology. We've done some work looking at the role of mitochondrial dysfunction in these abnormalities. It seems that the mitochondria are very susceptible to the in utero intervention. And we've shown in the liver, and we're doing some work now in the kidney with a group in Cambridge, that there seems to be uncoupling of oxidative phosphorylation, which can lead in itself to oxidative stress and to damage. But one of the more interesting sides of this, which certainly has relevance to the human condition, is that there is a reduction in nephron number as a result of maternal undernutrition, which is sort of, I suppose is reasonable to think. There's less nutritional availability, the kidney doesn't develop properly. And this actually happens in low birth weight children. They have a reduced complement of nephrons. And it's probably okay in terms of renal function until in adulthood, when you depend on renal reserve and having a bit of spare capacity in your renal function. And in fact, we think that this reduction in nephron number plays a very important role in development of hypertension in the offspring. There seems to be an increase in sodium reabsorption. What there certainly is in all these models is an increase in sympathetic efferent tone, which seems to contribute to the blood pressure going up, and that's through increase of renal sympathetic activity. And if we look at the genes which are involved, the reduction in nephron number seems to be due to apoptosis happening in the kidney through expression of these genes. And some people have shown altered methylation status of some of these apoptotic genes. So to give you an idea of what everybody's working at. But my interest actually has been entirely at the other end of the nutritional spectrum. When everybody was talking about growth restriction in those early years, I kept on saying, why are we so interested in maternal undernutrition when I see around me an awful lot of obese people? And I was a bit of a lone voice in those days, and we actually, my first PhD student who started doing this was a guy called Imran Khan, not the cricketer. But we set out with Clara Lowy, who was a diabetic consultant at that time, who was interested in the effect of diabetes in the mother. And we developed a model where we fed rats a high-fat diet, and we looked at the offspring, and we found that the offspring became hypertensive. Now, I went to one of these programming meetings, and David Barker said to me, I remember him saying, he said, Lucilla, programming is all about undernutrition. It's got nothing to do with the overnutritional side. And they gave me a poster presentation. I was very, very upset about it while having an oral presentation. But it is interesting now that probably about a paper a week is coming out with these models. Half the world has adopted the models, but we're pleased to say that we were the first people to do it. It's very nice to be able to do that. But the problem is that you know, obesity is a massive problem. We all know that. I was in Australia a few weeks ago, and one of the reasons for going to Australia was that there's a group working with us in a European collaboration. They've joined in, being Australians. But they're working on problems with obese pregnancies. They're doing a very similar study. I'm going to tell you about that in a minute to us. And we had a very interesting week comparing notes about our intervention studies. But you know, Australia is one of the epicenters of obesity now. But just look across the world. In South America, we used to you know, a developing country with relative poor nutrition. They're of course much more affluent now. And in countries like Chile, there is a huge problem of obesity. Alaska, the Eskimos, unfortunately, have now all got incredibly obese. It is such a huge problem. So back to the obstetricians in the clinic, this has produced a massive problem for them as well, because obesity in pregnancy, we're talking now about immediate risk, is associated with all sorts of awful problems. Not necessarily the case, and a lot of women have perfectly normal pregnancies, but because pregnant women themselves are insulin resistant, pregnancy confers insulin resistance on the mother. So if a woman is obese and already is insulin resistant, then she has a much higher risk of developing diabetes in pregnancy, and insulin resistance is associated with preeclampsia and so on. And then women are more susceptible to clotting, and postpartum hemorrhage is a real problem. We've just done a study of that, looking at the prevalence of postpartum hemorrhage, and it's shot up in the United Kingdom in the last few years, which is probably to do with the obesity epidemic. And then the babies, because of the increased glucose supply, which gets straight across the placenta, stimulates fetal insulin production, which is what's required for growth in the infant. The babies get fat, and then they get stuck when they're delivered, and then that leads to problems. Then the mother has to have a caesarean section. So in a study that we did with obese pregnant ladies, women with a BMI of over 35, about 50% of the women had caesarean sections. So you imagine the healthcare costs, and this is produced, any conference you go to now in the obstetric world, there's always a big session on obesity because it's become such a major problem. But of course, going back to the programming hypothesis, what we're interested in is the effect on the developing fetus. And there is now a very, very substantive literature. In fact, in some ways, it's much more convincing than some of the undernutrition work, which suggests that the endocrine milieu that the fetus is developed in, the high insulin, the abnormal lipids, and so on, is associated with an increased risk of that child developing obesity and diabetes in later life. I'm not going to go through it, but this is just some of the papers which have shown an independent association between maternal BMI and offspring BMI. People have now been looking at the relationship between maternal fat mass and offspring fat mass, and we're doing that at the moment. And independent, again, I'm saying these associations have all been corrected for all those confounders that people think may be responsible. And yet all these studies have suggested this association, and many more. This is just an example. What is big hot potato at the moment in the obstetrics world is gestational weight gain. You go to conferences on gestational weight gain, believe it or not. And the reason for that is that in America, everybody weighs women every time they see them. They've only got to look at them and put them on scales. In the United Kingdom, we only weigh them once at the beginning of pregnancy. And the reason for that is that we don't really know what to recommend to pregnant women as to how much weight they should put on. So there's a big transatlantic argument going on at the moment. But from our point of view, what is really interesting is all of these studies up here, and I've recently reviewed them, have shown that women who put on excessive gestational weight gain are far more likely to have children who are overweight or obese. And this is showing weight at 21 years, excessive weight gain being associated with greater incidence of obesity. Again, that's all been corrected. Some really beautiful studies published in really high-rate journals like Circulation and so on are showing now that this is an independent relationship. What we don't know, and this is where I differ from some of my colleagues who tend to get evangelical in this field. We know that childhood obesity is associated with the children sitting down doing nothing, watching television, stuffing their faces, doing all the wrong sorts of things to eat. We know that, for example, now that breastfeeding seems to be protective against obesity. We know that the hours that a child sleeps, if a child isn't sleeping very much, it's more predisposed to obesity. That may be because it just has more time to eat. And also community and demographic factors. are important. So what we don't know is what epidemiologists call the size effect of this in utero experience. And that is where we have to be extremely careful in making recommendations in terms of public health. And that's what we are all trying to do now, is to work out how much of this in utero influence is dependent, is causing childhood obesity. So one way we're approaching this, we're doing a big national trial at the moment, which is called the Upbeat study, and it's centred on all those places in England. And essentially, this is a study which primarily is designed to try and improve outcome in our obese ladies because of all those problems they experience. And what we've done is to develop a behavioural intervention. I've been working with psychologists for the first time in my life, and I've learned an awful lot. This is with the psychologists in University College, Professor Wardle and her colleagues. And they've helped us design an intervention which is based on psychological theory, where half of our ladies, women, are randomised to the intervention arm. They see a health trainer for an hour and a half on a one-to-one talk, and then they have 8 sessions with the health trainer where we try and change their behaviour in relation to their diet and in relation to their physical activity. Now, the dietary changes we're suggesting are based on a sort of anti-diabetic diet. This is really trying to prevent gestational diabetes. The primary endpoint of this trial is gestational diabetes. And so we're recommending they have low glycemic index foods and that they don't drink lots of sweet sugared drinks. I mean, most people when they're pregnant think that eating lots of fruit juice is really good for them because they should eat lots of vitamins, but it's absolutely the worst thing anybody could do because, as we all know, most of the orange drinks and so on are absolutely packed full of calories. So people come to us in the clinic with sort of 2 pints of cartons of orange juice and so on, saying, I'm being healthy, and in fact it's the worst thing they can possibly do. But so the primary outcome of this study is gestational diabetes, but of course from my point of view, we're very interested in following up with children, which is what we're doing now. Some of them are 6 months old. The first children will be 3 years next year. We've been doing 6-month follow-ups. And we have European funding. We've joined forces with a big European framework study to follow the children up until they're 3. And wouldn't it be nice to show that those mothers who adhered to this intervention, that they had a better outcome of pregnancy, also had children who were thinner when they grew up. Now, this isn't going to prove the developmental origins theory, but it is one of the best ways of doing it when you have a properly controlled randomised trial. So there's the first lady from Upbeat with her child. I hope this child was in the control arm of the study because that child is distinctly fat. So what about the mechanisms? The last few minutes I'm going to take you through, I'm running over time a little bit, but I'm just going to take you through what we've been doing in some of the animal models to try and tease out what's happening. What is really, I think you'd all probably tell me this is probably due to maternal hyperglycemia and that's the end of the day. That's probably not the case. There's a lot of suggestion that maternal hypertriglyceridemia is involved in macrosomial development of problems in the child. And also it isn't just the child is fat and stays fat, because these children often become thin and then they get fatter again as they get older, so there's a lot more to it than that. The area that we're particularly interested in is the role of fetal leptin. And I'm going to explain a little bit about that to you first. But first of all, before I talk to you about that, this is a study from my mate's laboratory in Harvard, Matt Gilman, who I've just written a book with, who I've spent a lot of time working with, where he has shown, for example, that mothers who eat a diet which is high in polyunsaturated fats have thinner children than those women who eat a diet which is rich in saturated fats. And look what this guy's managed to adjust for. He's done a really, really good study. He's measured everything he could possibly measure in the mums and the children. And this project, which is called Project Beaver, is probably one of the best cohort studies around in terms of what has been measured. So what are we doing in terms of our animal models? So we have developed this model of a fat mouse or a fat rat. I hope you like my graphics. That's the ears and the whiskers. And we've basically done this for years now, and we've been looking at the phenotype of the offspring. The offspring of these obese animals become fat, and they also eat more, which is important. They're also hypertensive, they've got altered liver function, they behave very strangely, And they get diabetes in later life. So there's no question about this, that you just make the mothers fat, that the offspring grow up very abnormal. But I'm going to take you through one very nice little story about this, which I think is absolutely fascinating. I said we're very interested in fetal leptin. Now, leptin comes from adipose tissue, as I expect you know. It's a product of the OB gene, and it is involved in satiety control. So normally, if we produce leptin, we stop eating more, which is a good thing. But unfortunately, if you become obese, you become leptin resistant, so it all goes a bit pear-shaped. But leptin is normally there to stop us eating, and it's produced in the adipocytes. It gets across the blood-brain barrier, and it affects the hypothalamus. So here is hypothalamus with all its different nuclei. Just to remind you, that's the arcuate nucleus. That's where the hypothalamus is, somewhere around your ear. And when leptin is produced, it binds to and activates centres in the arcuate nucleus which controls satiety. I won't go through into the detail. But there's more to it than that. In young animals and in late gestation in primates like us, leptin is doing something else. Leptin is controlling the development of neurons from one nucleus to the next. It's a growth hormone. It's a neurotrophic growth hormone. And if it's not there, everything goes wrong. And there are a lot of studies which show that. And here there's a nice little study in a dish. Here's an arcuate nucleus from an animal which is deficient in leptin. And if you add leptin to it, you can see all the neurons growing out. Really nice study by Sebastian Brewer and his colleagues. So to cut a long story short, we wondered whether there was something going on with this leptin story in our animals. So we measured in early postnatal life, during the suckling period, the leptin concentration, the red bars here, in the offspring of the obese animals. And this is the normal trajectory in controls. And it's this biphasic surge which is thought to control normal development of the hypothalamic neurons from one nucleus to the next. So it's very important for normal development. We found that the offspring of these animals are resistant to leptin pretty much when they're born. So we normally develop leptin resistance when we become obese. So if we give a normal animal, just 30 days old, that's one week post-weaning, we give it leptin, the 24-hour food intake goes down, which you'd expect, and the animal loses weight. That's what would happen in you and I. But in the offspring of these obese animals, that doesn't happen. They've already been born leptin resistant, which is bad news. We then looked at the hypothalamus of these animals. This was done in Clive Cohen's lab, and we found something very abnormal was going on. Now, essentially, what we looked for was AGRP neurons. Now, these come from the arcuate nucleus, but we looked for them in the paraventricular nucleus. And they're supposed, in normal animals, to get from the arcuate to the paraventriculum, who they all are. But in the offspring of the obese animals, that didn't happen. So this neuronal connection is not happening between the nuclei. And what we think is happening is that because there's so much leptin around, the brain becomes leptin resistant, and the whole neurotrophic development side of leptin's action goes completely pear-shaped and wrong. And we have good evidence that that's the case. So what we've done recently is to give leptin to young pups of normal animals and to try and mimic the leptin surge. Hasn't quite mimicked it, a bit higher, but we've done that. And to cut another long story, very short, these animals grow up with all of the features of the offspring of the obese animals. They become leptin resistant, they become fat, they become hypertensive, and so just by giving leptin in early life, we've created that offspring phenotype of fatness and hypertension. We think this is quite important. We're just about to send this latest paper off for publication. But what it is suggesting is, I'm not saying it's the only mechanism, it's suggesting that the brain and development of the brain is incredibly important in these programming models. And we are not the only people to show this. There are now probably 50 or 60 papers showing the plasticity of the developing hypothalamus in models of developmental programming. And in fact, the hypothalamus is known for plasticity in adult life as well, so this is a recognised phenomenon. Probably incredibly important. Hyperinsulinemia may be doing the same thing, we don't know, but we focused on leptin. Is there anything which might suggest this is happening in people? One study relatively recently measured cord leptin in women, diabetic women, and the same thing happened to the offspring of diabetic women who are obese, and showed a relationship between BMI at 7 and leptin in the cord blood. So there is some suggestion. Of course, most people haven't looked for it, and that's what we're doing now with our UpReach study. So I'm just going to finish with another interesting aspect of this model, which has really intrigued our gastroenterology consultant friends, and that is that the offspring develop something which looks incredibly like the disease called non-alcoholic fatty liver disease. This affects 17 to 33% of our population, particularly amongst people who are diabetic, particularly amongst people who are obese, and it can lead to all sorts of nasties like cancer of the liver, and it's associated with metabolic syndrome. So, and it's very prevalent. And just to show you, here are the offspring of normal animals which are fed a sort of chunky Western diet. So they get fatty liver. This is a normal animal. And this is the offspring of the obese stam fed a Western diet as well. And look how worse the condition becomes. This liver has been primed to develop non-alcoholic fatty liver disease. And working with our clinical colleagues who are interested in this in people, we're looking at mechanisms, and we found all sorts of mechanisms which lead to fibrosis and inflammation. The cytokines are increased, and the Kupffer cells are abnormal, and so on. And now these guys are using it as a model of non-alcoholic fatty liver disease. They're not really that interested in the programming bit, but it's a very good model. Okay, so I'm going to finish there. I hope this gives you an insight into this new hypothesis. Well, not so new, but perhaps new to most of you. What we're suggesting is that we have to be so careful about nutrition in utero, have to be extremely careful about trying to get people thin before they get pregnant. That's the best thing to do. And that otherwise we are probably going to be faced with a huge problem in later life, about which we can do not an awful lot because there has been this programming in utero. Thank you very much indeed.
Professor Poston, thank you very much. We've got time for a few questions if any of the students or staff would like to ask Professor Poston a question or two. Yes, here's a question here.
Just a lovely lecture. Thank you very much indeed. I found it very, very informative. Which part of a 9-month pregnancy is the most important in terms of nutritional underachievement, shall we say? If you think about the hunger window, that might have lasted 3 or 6 months.
Yes. There's good evidence for undernutrition at the very earliest first trimester is very important. But there's also evidence for third trimester in terms of growth being important. So there are lots of studies in sheep looking at different periods of deprivation. Each one does something different. But the early period when the embryo is implanting in the uterus seems to be particularly vulnerable. And we've also done some studies with the obesity model looking at the early embryo, which sort of lights up with oxidative stress markers and all sorts of problems there in terms of mitochondrial function. So from the earliest stage and different periods of exposure to essentially different things.
Most people see an overweight mother with an overweight child and say, well, she's overfeeding herself, but she's overfeeding the child. Have there been studies looking at children who've been adopted to see what happens to their weight when they go into a family that's not obese?
No, people have asked me that, and I'm not aware of any study, which would be a good study to do, probably because of numbers. But certainly the mother controls the diet of the family, and of course there's a huge influence of what she's eating is going to be reflected in what the family are eating. So there are all sorts of studies where people are looking at the family environment, the maternal diet, but there is a lot of sibling difference. And if you often see this in families, you'll see one very thin child and one rather fatter child, brought up in the same environment, and there's some pretty good evidence, for there being a direct in utero effect on one child and not the other. There's some very nice studies in siblings, which have shown, for example, that in utero exposure to diabetes will lead to obesity in the child, but the sibling who is not exposed to diabetes in utero is thin. So it isn't all that, but it's something which is extremely difficult to control for, and that's why, and we were looking very carefully in our studies too, it's very difficult to tease out the relative component.
Yeah.
If there's that consistent early life, and dietary improvements, how do we become more leptin responsive?
Yes, you can certainly become thin, you become more leptin responsive, yeah, absolutely. Helen.
I was going to ask about identical and non-identical twins. I know the water the instruments like that follow those. Has any studies been done?
There have been studies done with twins, and Tim Spector here has done some studies with twins, and he thinks there's remarkable concordance for obesity. What I keep on saying to him is that actually they share the same in utero environment, so it's actually quite difficult to tease it out. But there are studies in twins which suggest that the smaller twin becomes more obese, and there's discordance between twins, so it depends which papers you read. Unfortunately, a lot of the twin cohort studies which have been done don't have birth weight in the twin. So we are now actually starting a twin cohort study with Tim, where we're measuring, getting all the neonatal data prospectively, and we're particularly interested in developing brain. Actually, we're doing it with the Institute of Psychiatry, but we will prospectively collect the right data. I think largely the right studies haven't been done, so that's not.
Yes, I think.
I seem to remember there's a link between how much a grandmother eats and the
There is, yeah.
child degeneration rate. What part does that play?
There are several studies which suggest this goes beyond one generation. There's some very nice work from, certainly in animals, people have shown it going through 3 generations, but it tends to tail off in the 3rd, so it isn't consistent. That doesn't mean to say that the genes are affected, because you can imagine if the offspring of an obese mother becomes diabetic and obese, then she will have an influence on her child and so on, so it doesn't necessarily suggest that the DNA is persistently changed. There are studies in Scandinavia where they've looked at LGA, large deliveries in grandmothers, mothers, and the grandchildren, and it goes through generations in a way which you wouldn't expect from genetic association. So, yeah, there's quite a lot of evidence now for 3 generations, but not much beyond that.
Okay, one down here, Lucilla, down the front.
With infants, there's often the idea that you can't overfeed a breastfed baby, which is kind of a formula-fed baby. And I know there's been a change in recent years. Averages that people are trying to follow. So there's always this concern about dropping off the lines. And if you see babies, they don't develop straight. What role do you think current guidelines on feeding and not formula and calories has on the leptin?
There's the European consortium I'm working with is run by a paediatrician who's interested in just that. There's a lot of evidence that if a baby is born small and it's fed too much in the first few months of life, it's likely to become obese in later life. So that's a lot of work at the Institute of Psychiatry here. And of course, mothers, when a baby's born small, mothers really want to get their baby up to 50th centile or beyond. So there's always that. But very often, very small babies have to be fed on formula feed. And the study, which they've just done and continuing to do in Munich, has shown that if you reduce the protein content of formula feed, because it's a little bit high, if you reduce it, then the children go up with less adiposity. So the suggestion is that everybody's looking for the ideal composition of formula feed, and that study is suggesting that maybe it's just not quite right in terms of optimal development. So there's a huge amount of work going on there with especially the nutrition companies at the moment.
Does leptin have a role at that point as well?
Well, in women, I think, we don't know actually. It is generally thought in women that hypothalamic development is largely complete by delivery, but some of the studies in primates suggest it goes on a little bit longer. So we're thinking mainly 3rd trimester for pregnant women, and in rodents it's postpartum because they're born as altricial species and they're much more immature, so the hypothalamus is different levels of development. You have to be very careful about that with rodent studies, trying to equate what's happening in rodents to what's happening in mothers, and it's a bit of a problem. That's why it's better sometimes to work on sheep and primates.
Are there any evidence to suggest that late on in life, if there is no intervention on that person's sort of significant personal damage?
Not in people at the moment. There is in animals. So people have we're doing this too. So if you program an animal and then you reduce its diet and increase its physical activity, you can offset the problem. So that presumably would be the same. But the problem is we're in an environment of nutritional excess and plenty. These things may not be apparent unless you actually challenge the child with a Western type diet. So the suggestion is if you carry on the low plane of nutrition, this has certainly been shown in animals, under nutrition in terms of protein restriction in the mother, the offspring, if they're kept on the same level of nutrition as the mother, they will live longer. And there's a Nature paper from Cambridge about that, and they have reduced telomere shortening and so on. So if you stay on the same very carefully at the micronutrients in the mother as well as the calorific content of the diet. And there they've shown that the mothers are vitamin B12 deficient. Now, that is associated with alteration of the carbon-3 cycle and the methylation status. That's the sort of hypothesis there. And they get hyperhomocystinemia, which is associated with endothelial dysfunction and cardiovascular dysfunction. So, micronutrients, I haven't talked about that, and there is a bit of literature on that. So maybe, from population to population, different dietary components may be playing a bigger role.
I was just thinking in areas of the world where we're more likely to see
yeah, well, the Indian studies are the case because what's happening there is there's a terrific transition in nutritional intake from one generation to the next. And the work in India, a guy called Yannick's done this. He's been looking at between the generations where the mothers have been quite small and subject to undernutrition themselves, and then the children are growing up with a high nutritional status, and they get massively overweight. So a lot of the work that's been done has been done in countries which are undergoing that rapid transition of nutrition, and it's very evident in India. Yeah.
Well, Lucilla, thank you so much for that lecture. That was really inspirational, and our first Research Stars lecture. And I'd also just like to say thank you to everybody for coming to listen to our first Research Stars lecture, but it couldn't have been a better lecture, so thank you very much. Thank you.